All right, we will go ahead and get started. So today's lecture, we're going to pivot and talk about cellular and molecular biology as it relates to the immune system. So we'll be talking about cellular and molecular innate immunology. There are two arms of the immune system, the innate and the adaptive. And so we'll first be talking about the innate immune system. You guys will go on spring break and retain all of this information and hold it near and dear to your heart. And then we'll revisit and go to the adaptive immune system after that. And we'll be discussing innate immunology in the context of Crohn's disease. So Crohn's disease is a type of inflammatory bowel disease. Inflammatory bowel disease is a classification of diseases that are described as chronic inflammation of the GI tract. Underneath inflammatory bowel disease, there's typically two diseases that fall under that. This is more so for your benefit, not necessarily quiz material. I feel like I should sometimes caveat that before I kind of go on a soliloquy. So we have Crohn's disease and ulcerative colitis. They both differ in what segments of the GI tract they affect. Crohn's disease affects the entire GI tract. Ulcerative colitis, as the name kind of implies, it focuses on just the colon. The lesions in Crohn's disease can traverse the entire intestinal wall, leading to, we'll talk about this in a moment, fistulas, where it's basically like tunnels from one part of your GI tract to another. And the ulcerative colitis will just affect the mucosal lining. And so these two, even though these two different diseases kind of fall in the same category, they have vastly different phenotypes. And we're going to be focusing on Crohn's disease that covers the entire tract and the entire thickness of your intestinal wall. So the symptoms, because of its ability to affect your entire GI tract and can go across the entire intestinal wall, it'll promote malabsorption of nutrients. And so because of that, oftentimes patients who have Crohn's disease will have impaired growth, they'll have severe fevers and severe weight loss. In the GI tract, and so we have an image of a colonoscopy here, and oftentimes you'll have these abscesses in the intestinal wall, and these are oftentimes pus-filled lesions in your intestinal tract. Oftentimes, I'm sure everyone's kind of familiar with pus, but these are usually dead neutrophils that are pouring out from, or filling up underneath the surface here, and oftentimes will rupture and burst. It often leads to a lot of pain, discomfort, a lot of diarrhea. You'll also have ulcers, which are these open lesions on the intestinal wall. And oftentimes it'll lead to a fistula. A fistula is basically a hole in that organ that will lead to another cavity area. And so sometimes you'll have a fistula that goes from the lower colon, the anus, sometimes to the bladder, sometimes to other areas, but they're really, really problematic that can lead to really dire outcomes once a fistula is formed. There are other systemic symptoms that these patients have, anemia, and as well as some neurological issues because it is so systemic and it's a really severe inflammatory disease. So the prevalence, about 1.4 million people live with IBD, the higher classification of inflammatory bowel diseases. And typically for Crohn's disease specifically, the incidence rate is about 1 to 10 people per 100,000. So it has a fairly high incidence rate. And interestingly, it also has a bimodal distribution. What that means is if we charted, I'll do it down here, hopefully so everyone can see. If you charted age as number of cases as a function of age, you'll see two peaks where typically patients between 10 and 20 have it, and patients up to their 50s will present with it. And so and we're not sure why that bimodal distribution occurs, but we based on epidemiological work, we know that it exists. The etiology of the disease. So we talked before about monogenic diseases like sickle cell. Crohn's disease is considered a polygenic disease, that there are multiple genes associated with Crohn's disease, and that these multiple genes have mutations that enhance the susceptibility of a patient to develop it. And these mutations typically lead to malfunctions in the innate immune system. What ends up happening is that there's these malfunctions in the innate immune system lead to the inability for our innate immune system to function as it should, and it leads to an overcompensation of the adaptive immune system. And typically these symptoms are a result of a chronic and continuous immune response to microbial infections. But also in addition to that, as I'm sure a lot of you know, we have commensal bacteria, or we have a microbiome that lines our own intestinal gut, and oftentimes the adaptive immune cells will also respond to quote unquote our good bacteria that we have as well. And so we will talk about a gene that's associated with both innate immunity and Crohn's disease, and it's called NOD2. So kind of hold on to that for a moment, and we'll revisit NOD2 towards the end. But right now what we're going to do is we're going to try and describe the important qualities of our immune system. So this was already mentioned in your pre-lecture, but it's worth reiterating and revisiting. Our immune system has an incredible diversity to it. It can respond to a wide array of pathogens, and is oftentimes really important because there are so many different kinds of pathogens that exist and that can also evolve and mutate. And so it's important for us to possess a robust enough immune response that's diverse and that can tackle these different pathogens. What's also important is that it's also incredibly specific. The specificity of our immune system is accomplished by the ability of different cells of our immune system to respond to specific features, to a specific either epitope or a specific epitope on an antigen, or specific patterns that exist on either bacteria or other pathogens. And the last one is the memory. For a long time, we considered memory to be only something that exists in adaptive immunity, because those are the cells that secrete antibodies, and antibodies, as well as some of the cells, develop memory and last for years. And one of the ways that I've always enjoyed simplifying our immune system into innate versus adaptive, the innate immune system, I love the innate immune system. It's fast but dumb. The adaptive is slow but smart. That is the side that is typically considered to possess most of our memory. But there's a growing area of research that has been characterizing recently how we also have innate memory to some degree, that there are like repeated exposures to pathogens that some of our innate immune cells can kind of develop some sense of a memory by having changes in their cells. This is important, but this is, I think, what's cool about what's going on now. The immune system is constantly new things are being discovered about it. Its qualities and cells of it were discovered, over 100 years ago, but there's new cells being discovered every day, new qualities of these cells, new senses of diversity among these cells. And so one thing that's being discovered now is how innate cells also have a degree of memory. So we're going to break down the immune system even further. So we have innate immunity and we have adaptive immunity. Innate is considered fast but dumb, adaptive, slow but smart. Now we're going to break that down even further. We have the humoral immune response, which is kind of the what is present in our bodily fluids, primarily in our blood. So there's like the blood component of our immune response and the cell-mediated component of our immune response. So for innate immunity, the humoral response is characterized by a series of enzymes. We're going to visit that today. It's called the complement system, that can directly kill some foreign cells. The cell-mediated part are these cells that can engulf pathogens through phagocytosis. Phago eating, cytosis, kind of like entering the cell, so they're eating it, destroying it. For adaptive immunity, this is kind of going to be the last bit we talk about adaptive immunity today, because I want you guys focusing on innate immunity because it's so cool and elegant, even though I just described it as fast but dumb. The humoral immune response, humoral adaptive immunity, is characterized by antibodies that bind to and activate pathogens. And the cell-mediated part are going to be cells that respond to some of these specific pathogens that have been presented to them. So we're going to take a moment to look at the humoral innate immune response, which is primarily the complement system. Sorry. So there are 3 different pathways for complement. Complement is kind of similar to the coagulation cascade, a long series of enzymes that act on one another that lead to a final outcome, in this case, destroying and killing a pathogen. What I am not asking you to do is to memorize each and every single protein, what they enzymatically act on, what the product is. I am not asking you to memorize that, but rather to take a step back and to consider that there are 3 pathways that complement is accomplished, and maybe what is an important linchpin at the center of all 3. So we have 3 pathways. The first is considered what the classical pathway of complement. So that's the top left here. And what ends up happening is the bacterial surface is littered with antibodies. Now, it's possible that they could be the same antibodies that are across the surface of the cell, because there could be multiple of the same epitope across the entire surface of the bacterial cell. It can also be different antibodies, because different antibodies recognize different things, and the bacterial cell membrane is not a single antigen on it or one single epitope, it has multiple epitopes on it. So what's going to end up happening, step 1 for the classical pathway, is you have a bunch of antibodies across the surface of this bacteria. What ends up happening is there's this protein called the C1 complex. It's made up of C1q, C1r, C1s. What ends up happening is that they will enzymatically cleave other members of the complement pathway. C2 becomes C2a, C2b, C4 becomes C4a, C4b. But what the critical thing is, it'll lead to acting on one complement protein called C3. C2a and C4b together create this enzyme that acts on C3. Okay, so C3. So I want to hit pause on the classical pathway and point to that we're going to stop at C3 for that pathway. The key part of the classical pathway is that it begins with antibodies littered across the surface of the cell that then are recognized by the complement protein C1. In the lectin pathway, it's characterized by a lectin that's called mannose binding lectin. What's really unique about bacteria is that the sugars on the surface of their cells have a lot of mannose. Usually we express mannose on surfaces of different on different surfaces, but we do not express mannose as highly as bacteria. So I want to put like a virtual asterisk right there. This is one example where there's a distinction between our cells and bacterial. Mannose we don't, and our immune system evolved over time to recognize, okay, we should be recognizing mannose. So we have mannose binding lectin that binds to the mannose on the surface, and that will have other proteins that make this large structure. One of the cool things about the complement pathway, this is not important. The structures look kind of cool. It kind of looks like different spaceships docking on this bacterial cell. So mannose binding lectin with a couple other proteins form this complex that bind to mannose on the surface of bacteria. And what ends up happening is it'll elicit the conversion of C4 to C4a and C4b, C2 to C2a, C2b. And again, it'll lead to the production of this enzyme complex, C2a and C4b, that'll act on C3. So again, both of these pathways are converging on C3. They're creating these enzymes. It's called the C3 convertase. That's what C2a and C4b is, the C3 convertase, and they'll act on C3. So we'll push pause there. Now, as C3 is being converted to C3a and C3b, what ends up happening is we have this alternative pathway that becomes activated. It's kind of like an amplification loop. There's all of this C3 that's becoming C3a and C3b. And as you're getting more and more C3b, what will end up happening is you'll get C3b wedged, beginning to become wedged in the bacterial surface, and that'll activate the alternative pathway. And what ends up happening is that C3b becomes activated by interacting with endotoxin. That's these like little, we'll talk about that later, but they're like these little features on the surface of a bacteria. And so what ends up happening is the key thing to remember for alternative pathways is that C3b is activated by endotoxin on some of these bacterium, and it'll create another C3 enzyme. And so we have one way where the C3 is clipped by the formation of this new enzyme here. and you have C2A and C4B on the other pathways that act on C3. So this is a lot of information, but the key takeaway, to take a step back and look at this and think about what's going on here. All of these proteins, C1, C2, C4, these are in our blood. They're circulating. And for the classical pathway, when antibodies are littered across the surface of a bacteria, the complement pathway is activated by C1 complex binding there. For the lectin pathway, it's mannose binding lectin that recognizes the mannose on the surface. And the alternative pathway is like an amplification loop by some of the things that are being produced from the other pathways. Now, I think this is really cool. Some might not agree, but what ends up happening is that you make another enzyme for another complement protein called C5. And you make, as you can imagine, C5 becomes C5a and C5b. Now, there are three outcomes of complement. The first is that the enzymatic byproducts, C3a and C3b, become anaphylatoxins that recruit phagocytes to that location. All right. So that's one way that complement is working here. Another outcome is that, as I mentioned before, C3b is becoming littered on the surface of this pathogen, and our immune cells have receptors for C3b. So these receptors are activated, or these receptors become activated by C3b and trigger phagocytosis in some of these innate immune cells. And the last thing, I think this is really cool, last way it reaches an outcome is that it actually will create this complex where C5b joins with all of these other complement proteins, and it acts on C9 where it creates this pore. It's almost like drilling a well in the surface of the bacteria. And all of a sudden it disrupts protein, it disrupts the protein transport and the osmotic balance, and it basically lyses the bacteria. These are some of the ways that complement can act on a pathogen, where the byproducts recruit phagocytes. The C3b, one of the proteins that's activated, acts as a ligand for a receptor that triggers phagocytosis. And you'll actually have this membrane attack complex, the MAT complex, basically, that will drill a hole in this bacteria to blow it up, to lyse it. And so those are some of the ways that pathogens are destroyed by the humoral innate response of complement. Are there any questions there? Yes. Great question. So the question was, as I understood it, do they all happen at the same time, or other ones? Is that what your question was? Forgive me. So all of them can happen, or just two of them, typically followed by the alternative pathway. So you can have the classical pathway followed by the alternative. You can have lectin followed by the alternative, or all three can be happening at once. But the main takeaway is to think about what triggers them. So lectin is triggered by mannose binding lectin. The classical pathway is triggered by antibodies, and the alternative is an amplification loop that is triggered by one of the other two. Yeah. Yeah, so ends up happening, I was trying to figure out a way to include it here. The formation of C3b will lead to C3 plus 5. So you have the C3 convertase up here, C4, C4b. You have another C3 convertase from the alternative pathway that's different, factor B, factor D, and that leads to C3b, and then C3b with C3 makes a C5 convertase. I was trying to avoid focusing on the different proteins, the different convertases, because it's really easy to get distracted and focus on those. I can remember spending a lot of time trying to memorize each single one, how they bind together, and I don't want you to get distracted with that, but rather the big picture is that there are 3 pathways to complement, and they get triggered by different molecules, and that one of them, the alternative, is an amplification that helps convert more C3 to C3. And that ability to convert more C3 to C3b is important because it helps lead to the formation of this complex up here that then leads to the membrane attack complex, or C3a and C5a become anaphylotoxins that recruit phagocytes, and C3b on the surface of these cells can also trigger phagocytosis. There's a receptor and it triggers them. Are there any other questions? Yes. The latter. So what ends up happening, the name anaphylaxis means it's a toxin that elicits anaphylaxis. What that means is, I'll do it right here. Our blood vessels are there, and we have immune cells here that have receptors for C3a and C5a. When these immune cells get activated by C3a and C5a, again, this is not information that's you're going to be quizzed on, but just rather to answer the question. Some of them are mast cells, some of them are macrophages, some of them are, I think dendritic cells express this receptor. Anyway, but what happens is they trigger histamine release, they trigger cytokines and chemokines. We'll talk about what those are in a moment. It makes your blood vessels leaky so that it's easier at this location in your blood vessels for the immune cells to leave the blood vessels and to come to this site in the tissue, where then they'll be activated by C3b and undergo phagocytosis. They'll leave the bloodstream because of what C3a and C5a do, because of these other immune cells nearby, and will come specifically to that site to undergo phagocytosis. Are there any other questions? So I'm not asking you to remember that what an anaphylaxis is, what that is, but rather that there are 3 outcomes that can happen as a result of complement, and that the enzymatic byproducts, or what's coated on the surface, or the final product of complement, the membrane attack complex, where it's literally elegantly drilling a hole, and it's not just one, it's all over the surface of this bacteria, that these membrane attack complexes that are bursting this pathogen. Are there any other questions? All right. Yeah, cool.
Reading here, looks like it's most effective with the gram-negative bacteria. That's because of the thin wall layer there.
Sorry, you said what is? You said green?
Yeah, the MAC complex is most effective with gram-negative.
We'll get to that. Yes, hold that thought. Based on my drawings on the board, you're going to get to where I'm going to get hopefully soon. So try and move this out of the way. So that was humoral mediated immunity. Now cell mediated immunity. We have a series of receptors that bind a series of ligands. These ligands associated molecular patterns or damage associated molecular patterns. And they're called PAMPs or DAMPs. It's not an elegant name, but it's what we have. So PAMPs are recurrent features or patterns of innate immune cells over the course of evolution. So a lot of the immune cells that we have are in shellfish, are in lower order organisms that have been maintained over the course of millions of years of evolution. And in addition to that, we have DAMPs, these damage associated patterns, where during cell damage or during tissue disruption, there are these factors that are released that you can kind of imagine them as flare guns almost, that signal there's tissue damage here, we need to activate and recruit immune cells to help take care of destroying any pathogens that might lead to infection, or, and this is kind of the area of research that I'm in, how can those damps also trigger immune cells to promote tissue repair and tissue regeneration? And some of these damps, for example, are ATP, uric acid, or certain cytokines. They're usually stored inside a cell, but when released prematurely can serve as, again, that flare signal as a damp to signal tissue damage. Now, what's important to know is that PAMPs and DAMPs bind to pattern recognition receptors. So PAMPs and DAMPs, you can think of them as almost like a polymer. So just for clarification, there are a few slides here that I've added that I will post today as I post this, the video. So don't feel the need to take photos because I'm going to post this with the video today. Multiple people are doing it. I'm not just calling out specific people here. So the important thing to know is that PAMPs and DAMPs are bound, they bind to pattern recognition receptors. So some of the things that occur when PAMPs or DAMPs bind to a pattern recognition receptor is that they can elicit chemotaxis. What'll end up happening is, if you remember the pre-lecture video where a neutrophil is basically going around and chasing a bacterium, there's some sort of chemoattractant that bacterium is releasing. And so that chemoattractant is a PAMP, and it's trying to chase down and get to that bacterium and eventually phagocytose it and destroy it. And so the pattern recognition receptors binding to PAMPs or DAMPs will undergo chemotaxis. Chemotaxis is traveling in response to some sort of molecule, some sort of chemoattractant. You'll also get anti-pathogen defenses. In addition to that, you'll get the secretion of cytokines and chemokines. So this is important. A cytokine is a protein that is typically released, some soluble factor released by and communicate with other immune cells to activate them and to trigger specific responses. It can also be released by other cells, non-immune cells, to communicate to immune cells to tell them to become active. And so different cytokines elicit different responses, but it is a way of immune cells to be communicated with. There are some cytokines that trigger chemotaxis, that trigger the movement of that immune cell to a certain place. Those are called chemokines. And so, as you can imagine, these cytokines and chemokines, because they're eliciting specific actions, they also elicit changes in gene expression of these immune cells. So to take a second to think about the big picture here, we have pathogen, as the name implies, pathogen-associated molecular patterns. These are almost like polymers that have a specific pattern that's on a pathogen, that's on a bacterium, that's on a parasite, that's on a virus. These are PAMPs, they're pathogen associated. We also have damage associated as a response to cell damage or cell injury. They bind to pattern recognition receptors. So these specific receptors recognize specific patterns. And it's been evolved over time to recognize specific patterns that are associated with specific pathogens. So we're going to talk about a few of them there, but I'll stop here for a moment and ask, I want to have a few questions. Are there any questions about what we talked about with PAMPs, DAMPs, and PRRs, pattern recognition receptors? Yes.
Not directly separated, but you said cytokines were the ones that cells that tell them to be active and whatnot.
Yes.
And then
Yes. I want another question. Yeah. That's a great question. So pattern recognition receptors expressed on immune cells. Typically, we only thought of them as being expressed on the surface because, bacterium or viruses are out there. But as we've seen with the serial lecture, where it's a bacterium that escapes the endosome and circulates around, as we've seen with coronavirus, they are cells that enter into our cells. We also have pattern recognition receptors that are intracellular. They are in our cells. They are in our endosomes. They are situated across different parts of the internal side of our cell. We're going to look at some that are extracellular and look at some that are intracellular. I also really like dumb questions, in case I haven't said that already. And if something doesn't make sense to you, that's not your fault. That's my fault for not communicating it clearly enough. Ava. Yes, they are the ligands for the receptor, pathogen pattern recognition receptors. So PAMPs, pathogens, they come from other things. DAMPs come from our own body, from our own cells. Yes. Host encoded. What that means is they are in a lot of, that's a very good question. Innate immunity is something that we are born with. It is encoded in us. Adaptive immunity is acquired. It's all historically adaptive immunity is also called acquired immunity. You are acquiring immunity against things that you're seeing and exposed to. So all of these innate immune responses, whether it's the human response with complement or the cell-mediated response, these are encoded in our DNA. So we express certain receptors, we express certain DAMPs, important in our ability to quickly respond to pathogens. So like I said, innate immunity is fast but dumb because they're not responding, they're not remembering and responding to like area specific, like several amino acid epitope peptides. Rather, they're generally responding to these patterns on things that we've seen over millions of years of evolution and have been encoded in us to remember them. Yeah, I'll leave it. All right, I'm gonna keep, go ahead, Lauren. Today, a very important question for how adaptive immunity works. It's also fairly complicated, but again, we'll keep it at a big picture to understand how it works, but hold on to that during your spring break. Okay, one extracellular pathogen recognition, pattern recognition receptor we have is the formyl peptide receptor, FPR. So it is a family of G protein coupled receptors that recognize formylated peptides. All right. So typically when we have, typically during translation, most of our proteins begin with a methionine. However, and again, I had that like virtual asterisk that I asked you to remember, like how our host cells and bacteria are different. During translation, during the production of proteins, we have something called a start codon. It's AUG. You can think of school starts in August, AUG. And that typically is methionine. For bacteria, it's a formylated methionine. And so we have FPRs recognize formylated peptides, and in this case, formylated methionine, leucine, and phenylalanine. And so that's the classic ligand for this formyl peptide receptor. And so it is a powerful chemoattractant for neutrophils and macrophages. And what's really interesting and noteworthy is that there's 3 FPRs that are encoded in humans and 6 encoded in mice. Now, given that this is a receptor that recognizes a ligand on bacteria, why do you think we only have 3 and mice only have 6? You know, typically, this isn't meant to denigrate mice. We are typically considered a higher order organism, but why would they have 6 instead of us only having 3? Can anyone think of like an evolutionary reason why they would have more? Yeah. Sorry, I couldn't hear very well. You're circling around the right answer, but why, in the absence of medical interventions, think about our lifestyles, us compared to mice. Yeah, we're cleaner than mice. They live in the trash. I hope none of y'all live in the trash. but mice typically have a much dirtier lifestyle. And then there's an evolutionary need for them to express more to recognize different formylated peptides. Are there any questions there? So again, this is a slide where it's easy to be distracted by a chemical structure, easy to be distracted by memorizing methionine, leucine, phenylalanine. But the big thing here is that there is a difference in us versus mice where we do not have a formylated methionine, and they do during translation. All their methionines are formylated. And so that is one way that our immune system has evolved to recognize bacteria in that way. Another set of extracellular transmembrane, both actually, forgive me, transmembrane and intracellular pattern recognition receptors are called toll-like receptors. So these were, so traditionally, fly geneticists are usually the ones who historically have discovered things first. These were first discovered in flies, and they named that gene toll. Then it was discovered in people, and for lack of a better name, they were called toll-like receptors. And so toll-like receptors are a huge, important family of pattern recognition receptors that are expressed to different degrees across all immune cells almost. And so some are at the cell surface, some are intracellular. Depending on what they recognize, that'll dictate where they are. So for example, TLR3 recognizes double-stranded RNA. So just to make sure I say this out loud, I'm not asking you to memorize this list, but rather that this is one group of pattern recognition receptors, and that where they're expressed is oftentimes dictated. So TLR3, double-stranded RNA, that is only in a virus. We have single-stranded RNA during transcription, but a virus has double-stranded RNA. And so thinking about how a virus acts, if it's intracellular or extracellular, where do you think TLR3 is expressed? Intracellularly or extracellularly? Intracellularly. So TLR3 is an intracellular toll-like receptor. TLR4, the ligand for that is lipopolysaccharide. It's for gram-negative bacteria. As you can imagine, most bacteria exist extracellularly, so that is on the surface, and that is a really important toll-like receptor for macrophages. So to take, actually, I'll keep going. TLR5 recognizes flagellin on some bacteria that are motile, and so TLR5 recognizes the flagella portion. And lastly, TLR9. So in segments of our DNA, where there's lots of C and Gs, if it's a C and G rich part of our DNA, we call those CpG islands. And it is typically methylated for us. In bacteria, it's unmethylated. So this, despite it not saying here, TLR9 recognizes unmethylated CpG islands. And so again, and also what I should say is that TLR9 is intracellular because when we would see the DNA of a bacteria, it's inside and being enzymatically cleaved and lysed, so we can access their DNA. So LPS, lipopolysaccharide, is the ligand for TLR4. And so it has this glycan polymer at the top with a core oligosaccharide and these lipid A tails embedded in the cell cell wall. And this lipopolysaccharide, or LPS, is also referred to as endotoxin. It's called an endotoxin because if our immune cells are exposed to an infinitesimally small amount of LPS, it will trigger a really severe and robust immune response. And so whenever medical products are being developed, they are required by the FDA to be endotoxin free. And in addition to that, typically bacteria that express LPS at the surface are considered gram negative. So some of you may already know this, some of you may not. There was a microbiologist a long time ago named Graham, who developed this stain to try and classify bacteria in two different groups, whether they took up this stain or whether they did not. And for the different stain, or for the different groups, I have illustrated their structures on the board here. Typically for a gram negative, they have two cell membranes, and in between that cell membrane, those cell membranes, they have what the stain is meant to target, peptidoglycan. It's basically sugars with like a peptide, with an amino acid groups with it. And so it's impermeable and unable to access the peptidoglycans that are in between the cell membranes. So these are gram negative. And gram positive bacteria, they have a peptidoglycan surface around it that also possesses lipoteichoic acid, as shown on the slide here. And behind that, they have a cell membrane. And so the gram positive bacteria are able to take up the gram stain, whereas gram negative are not, because of their membrane structures. Because for gram negative, peptidoglycan is embedded within two plasma membranes or cell membranes, and gram positive, it's not, it's just on the surface. And on that surface is lipoteichoic acid. That's a polymer with these sugars that has a DAG anchor into it, embedding it in the wall. Yes.
So the bacteria there, the little red,
Yes.
those are the organs that
That exactly. Yes.
so this acid is embedded in the
Yes. Yeah. And so this is the ligand, LTA, lipoteichoic acid, is a ligand for TLR2. And so we have different receptor, toll-like receptors, or TLRs, that bind to gram negative versus gram positive. Yeah. I think I'm blocking up the gram positive label here. No, you're good. That's on me. I'm using Zoom to try and record these lectures now because or another recording software. But lipoteichoic acid is a gram positive bacterial cell wall component. Are there any other questions? Yeah. Gram negative is TLR4 because that recognizes lipopolysaccharide. That's part of that outer cell membrane there. All right. So as I mentioned before, another ligand for our toll-like receptors is for these CpG islands, these cytosine phosphate guanine islands. And so different segments of DNA are CpG rich or CpG poor. But the important thing to know here is, again, asterisk, our CpG islands are methylated, whereas bacterial CpG islands are not methylated. And so TLR9 recognizes unmethylated CpG islands. And these are just repetitive stretches of cytosine and guanine that are next to each other. And so it's just a normal part of biology where sometimes they're present, sometimes they're not. But the key takeaway here is that our bacterial DNA, that when exposed within the endosome because they're being lysed open, TLR9 is present in the endosome and in the lysosome. And so it's a really unique or a really elegant system where they're lysed, and TLR9 is right there in the endosome or the lysosome to recognize if there's unmethylated CpG islands. And if it is, then TLR9 will activate downstream pathways to communicate there is a bacterium here. We need to become more inflammatory to attack and kill whatever bacteria are out there that are making their way in through the endosome or the lysosome. Are there any questions? All right. second to last, forgive me. The second to last intracellular pattern recognition receptor we're going to look at is called cGAS. It senses cytoplasmic DNA. So if you remember, I've kind of referred to this a couple times now. There are bacteria that make their way in, and when we're lysing them, we should not have, I'll take a step back, asterisk again. Should we have DNA in our cytoplasm? No. So if there is DNA in our cytoplasm, it's not our DNA. It is from some sort of foreign pathogen. And so cGAS senses cytoplasmic DNA, and it generates what's called cyclic GAMP. It's actually taking cyclic GMP and cyclic AMP and GMP and AMP and cyclizing them together to make cyclic GAMP. And so what ends up happening, that's really important here, forgive me, oh my goodness, is cyclic GAMP activates a protein complex called STING. STING is really important because it leads to the expression of a family of proteins called interferons. Interferons are really important because they are, once they bind to their receptor, interferon receptors, there's different classes of interferons, but once they do, that is a really powerful way for our immune cells. There is something going on inside this cell. We should either kill what's inside it or kill that cell. And so the key takeaway here is that cGAS senses cytoplasmic DNA, which should not be there. It generates cyclic GAMP, and that activates STING. If you were to Google STING or to look it up on PubMed or Google Scholar, because I'm sure all of you have PubMed and Google Scholar now bookmarked on your web browsers because it's so important for everything we're doing. STING is it's a really powerful inflammatory pathway that triggers interferon expression, and interferon is a key protein in eliciting robust immune responses. Are there any questions? I'll wait for a couple of questions. I'll put it that way instead of asking if there are any. Yeah. Yes, great question. All right. Another intracellular pattern recognition receptor is called NOD. So NOD recognizes bacterial peptidoglycan in the cytoplasm. So as I mentioned before, there is a segment of bacteria that have peptidoglycan either on the surface or embedded between two membranes. And we should not have peptidoglycan inside our cytoplasm. And so NOD2 specifically binds to an element of a segment of peptidoglycan that's called muromyl dipeptide. It's called muromyl dipeptide because it has muramic acid up here with two amino acids down here. And so it is typically expressed in monocytes, macrophages, and gut epithelia. And so the gram-positive bacteria that express peptidoglycan on the surface are typically considered bad for our gut because we'll activate NOD2 activation in our monocytes, macrophages, or in the gut epithelium. And so taking this back to Crohn's disease, NOD2 is considered a susceptibility gene for Crohn's disease. There were 2 studies done by different groups that were looking at trying to identify specific genes that were implicated in Crohn's disease. And in the same, I'll move this over so you can see, in the same issue of Nature, published on back-to-back pages in the same issue, there was one group that used what's called a linkage analysis. They're trying to basically link certain mutations and regions of the DNA with this disease. And what they're able to identify is that in chromosome 16, there's what's called a susceptibility locus there. Locus means a site in the DNA or a site in our genome. And that site of the DNA or our genome had an elevated linkage or an elevated association with Crohn's disease. And so This group published in Nature 21 years ago that in chromosome 16, there is a locus of that chromosome that is associated with Crohn's disease. The next paper, right after that, another group looked at a specific family where the mom and dad were heterozygous for a specific phenotype. What this phenotype was a truncated NOD2. Now, their children both had a truncated, they were both homozygous and inherited this truncation. Mom and dad were both heterozygous. That means they had one copy of this truncation. And as you can see from the DNA gel here, the parents are in lane 1 and lane 4. They had one wild type allele, which is the normal version of NOD2. And they had this mutated form called 320 insert C. And so mom and dad had one copy of it. The kids inherited each of the mutated copy and only have truncated NOD2, which is a loss of function. As the name implies, loss of function means NOD2 does not work anymore. And so these two studies back-to-back identified that at chromosome 16, there is a susceptibility locus there. NOD2 is on chromosome 16, and that there's a truncation that leads to a loss of function in NOD2. To take it a step further, one group took that mutation, the NOD2 320 insert C deletion, and they knocked it into a mouse. So a knock-in mouse is when we take the embryo, the blastocyst of a fertilized fetus from mice. In this case, we are knocking in some sort of mutation, and these mice have that same mutation that these kids had in the previous slide. And looking at chemically induced colitis, chemically induced inflammation of their intestines in these mice, we're able to see is that WT, the wild type normal allele, only had a certain response to it, but in the knocked-in mice that carried that mutation, their colons were far more inflamed and had far more fibrosis or scar tissue formation in response. And so this mutation seemed to have a really robust and effective ability to induce colitis as compared to a normal allele wild type mouse. And in addition to that, this was no different than if you completely delete the NOD2 gene. So that small mutation had the same phenotype as completely deleting the gene itself. So for treatments, corticosteroids, they reduce inflammatory gene expression. In addition to that, antibody neutralization for specifically tumor necrosis factor, that's a really powerful inflammatory cytokine. And these antibodies neutralize TNF. And one of them you might recognize, Humira, is the #1 drug in the world because so many things are associated with TNF expression. Finally, if you're interested in innate immunity or Crohn's disease, these are the top rows, a group of people that are here at UVA that do fantastic immunology work. And the bottom two are people, I should move this so you can actually see his name, are two fantastic immunologists at different universities that also take on summer researchers all the time to include people in their group. But I appreciate you guys coming. I appreciate you guys sitting through this, and I'll be here if you have any questions. Thank you.